Dear students, I hope you are all well and studying in your home. Today we will start our new chapter number 4, Transpiration. In this chapter, your syllabus is Process and Significance of the Transpiration. Experimental work includes the loss in weight of a potted plant or a leafy shoot in a test tube, the use of cobalt chloride paper, Ganong's potometer and its limitations, the factor affecting rate of transpiration. So, what is the scope of your syllabus? So, you have to uh, learn and read about the mechanism of stomatal transpiration. Okay, and you have to clear your process thinking of that stomatal transpiration and adaptations in plant to reduce transpiration to be discussed. A brief idea of condition and bleeding should be given. Okay, so this is by means of your syllabus. So now we start the chapter. Plants absorb large quantities of water as well as mineral salts from the soil with the help of their roots. This water is conducted in the upward direction up to the tip of stem branches and leaves by a process called ascent of sap, as we already read about the last chapter. From the absorbed water, only 1 to 2% is utilized by the plants for the purpose of photosynthesis, only 1 to 2%, and other processes. The remaining 98 to 99% water is lost by the process of transpiration. Through the exposure of surfaces of plants, major parts of the plant water is lost by way of transpiration from the leaves, which have large surface area. Okay, as we know that in xerophytic places or in desert, the leaves are converted into spines to reduce the process of transpiration. So in tropical forest, mainly you can see where there is more amount of rain occurs, so their leaves will be very broad in compared to the desert leaves. Transpiration is very useful for plants for the two reasons. One, to get rid of excess water absorbed by roots, about 98%, and two, for cooling the plant in hot weather. So there are two significance of this process for a plant. The process of transpiration. Now you have to learn about the process, how transpiration take place. The loss of water in the form of vapors from the aerial exposed part of the plant is called transpiration. Okay, loss of water in the form of vapors from the aerial exposed parts of plant is called transpiration. It can be shown with the help of the following experiments. Okay, there are some experiments also given on your course book. So first experiment is to demonstrate the process of transpiration. Procedure: take a small well watered potted plant. Okay, cover the external surface of the pot and its soil with oil. Okay, you can see in figure also, like that you have to do. Place the potted plant on a glass slab. Invert a dry bell jar over it. Seal the edges of the bell jar with the help of Vaseline. It will prevent the entry of outside air to the bell jar. Okay, so this Vaseline work is to stop the entry of outside air to the bell jar. Leave the apparatus undisturbed for some time. Okay, you have to leave the apparatus as it is without any disturbed for some time, or cover the plant with a polythene bag and tie mouth around the base of the stem. So you can see in figure, you have to do this process. Okay, outside the pot you have to put Vaseline. Okay, this is the glass slab. Outside this bell jar and a potted plant inside it. Okay, so now what you have to do, you have to absorb. Soon the interior of the bell jar or polythene bag becomes misty. Afterwards, drops of water appear on the inner surface of bell jar or polythene bag. So you can see here are the some drops of water vapors. So what is the inference of or conclusion of this experiment is the water drops on the interior side of the bell jar or polythene bag can neither come from outside air nor from water present in the soil. The pot, because both have been properly sealed, the water vapors can come only from the exposed aerial shoot of the plant. Such a loss of water from the aerial parts of plant is called transpiration. Okay, so we know that no water is coming from outside and no air is coming to inside of the bell jar. So this water comes only from the shoot part of this plant. So this process is called transpiration. There is another experiment to demonstrate the process of transpiration. Procedure: take two well watered potted small plants, preferably with broad leaves. Cover the pots and soil of the pots with oilcloth. It will prevent the evaporation of water from the soil of these pots. Now, arrange three setups as follows. Setup A: take one of the above potted plants and place it on a glass slab. Cover the entire plant with a bell jar. Seal the mouth of the bell jar with Vaseline so as to prevent the entry of outside air into the bell jar. So figure also given 4.2a. So you can see pots covered with oil cloth. Okay, and this is a bell jar and inside it a potted plant. Setup B: place the second potted plant as in setup A, but also place a dry cobalt chloride paper inside the bell jar. So you have to one modification have to do, that cobalt chloride paper have to put inside the bell jar of set B or second plant. Setup C: as in setup B but without a potted plant. So in setup C, no potted plant inside it. Okay, you can see. Now, observation: the bell jar becomes misty after some time. Water drops appear on the inner side of bell jar. Okay, these are water vapors inside the bell jar and it become misty. It also shows the same changes as in setup A. In this setup, the initially blue colored cobalt chloride paper also turned pink. So you can see here it changes into this pink color. Okay, no water drops appear on the interior part of bell jar and there is no change in the color of cobalt paper. Okay, in setup C there is no changes will be occur, but here it will be colored will be changed into pink. Okay, so these things happen. Now how can you interference or conclude this? Condensation of water drops on the inner side of bell jar of setups A and B and change in the color of cobalt chloride paper A in setup B gives a double visual proof of transpiration. There is no source of water drops in these setups other than loss of water from the exposed part of potted plants. In setup C, no water drops appear on the interior of the bell jar. The color of cobalt chloride paper also does not change because there is no plant which could release water vapors by transpiration. Setup C represents control experiment. Okay, so now you can understand that the process of transpiration. So there is something about the control experiment. It is the setup which used to comparing the result is called control. It means in this actually it run along with the actual experiment, but in this setup one or more factor plant and the other things will be covered the pot. Okay, under study is or eliminated. Okay, whatever you do in this A and B potted plant you not do in C, so that's why it is come in the control experiment. So there is some fact about, did you know loss of water due to transpiration is about 2 liters per day in sunflower, 36 to 45 liters per day in apple, and 1 ton per day in elm tree. There are some difference between transpiration and evaporation. First is It is water loss from the surface of the cell, and it is the loss of water from the free surface of water. Okay, it is loss from the free surface of water, and it is loss from water of the surface of the cell. Okay, and it is a physiological process, and it is purely a physical process. It involves living tissues, it involves non-living matter. It is controlled by such processes as osmotic potential and water potential. It is not controlled by any such process. Light, CO2 concentration, pH, and hormones regulate and control transpiration. They do not influence evaporation. It is comparatively a slow process. It is faster. It is influenced by the anatomy of the transpiring organ. There is no such influence, and it is regulated by the amount of soil water available to the plant, and it goes on unchecked as long as water is present. So there are some difference between the process of transpiration and evaporation. From home, you have to read about the measurement of transpiration. If there is any confusion in this experiment, whether given 4 or 5 experiment for that, so you can ask me. Okay, and if available, I try to make an animation video for this process of transpiration. Okay, now we will discuss other things in the next class. Thank you.
So we'll talk about what transpiration is, what's good and what's bad about it, how it actually works, and how it actually enables the water to be transported across the plant. So, and then later on we'll also talk about how we can actually estimate the rate of transpiration and what are the factors that will affect it. So imagine here we've got a plant, so we've got a flower with the roots underneath here, and the roots would absorb the water from the soil. And then we've got the leaves here, the xylem going up and into the leaves, and in the leaves we've got different cells. So imagine that these blocks are talking about the cells in the leaves. Transpiration is the loss of water vapor by evaporation through the stomata. It's worth noting and underlying 2 words here, which is the word vapor and evaporation. A lot of the times that people will lose marks in the definition of transpiration by saying that transpiration is loss of water. That is not correct, because water vapor is considered to be a gas, whereas water itself is a liquid. So if you say the loss of water, that's not correct. It has to be the loss of water vapor by evaporation for the stomata to be the full definition for transpiration. So make sure you make a note of that. There are several different things that this is good for. So first of all, transpiration is kind of like sweating in plants, so it cools the plant down. So by the loss of the water vapor, it's because they absorb the heat and it turns into kinetic energy, and they just kind of diffuse across the cells and out of the stomata itself. Another good thing about it is that it actually causes the transpiration stream or the transpiration pool. And the transpiration pool is basically the movement of water and mineral ions up the stem, up the xylem from the roots. And because of the loss of water vapor here, it will cause that to happen. So imagine if the water vapor originally was in this cell and it's near the stomata, and because of various reasons, because of, for example, these ones, because it's hot, the water vapor will move out of the cell, becoming water vapor, and diffuse out of the stomata. And because of that, that will cause the water potential here to decrease. And in comparison to this cell here, which has a higher water potential, the water in this cell would then be drawn to the cell that has just lost some water. Now, if that happens, that means this cell, originally having a higher water potential, will now have a lower water potential compared to the cell next to it, which has got a higher water potential. And therefore, again, the water will then move from this cell to the next, because this cell has just, in order to replace that water that has just been lost by the middle cell here. And this kind of carries on and happening until it reaches xylem, and it's about that pool of water from the root up the stem through the xylem. So that is the transpiration stream. However, there is a downside to transpiration. Transpiration is about the loss of water vapor by evaporation, and that simply means that the plant is losing water. And if the plant is losing water in the terms of a single cell, the cell would lose turgor pressure, meaning that it will become flaccid or even plasmolized. And if it becomes plasmolized for a long time, then the whole plant will start to wilt, kind of like the leaves will curl up and the whole plant just kind of strengths a little bit. And that idea is actually as a protective mechanism, is to about trying to reduce the transpiration from happening, or reduce the rate of it anyways, to stop any further loss of water. And if the plant doesn't gain extra water during that time, then eventually it will die because of the lack of water to enable the plants to stay upright and to actually do photosynthesis. So that is the downside to transpiration. Sometimes we say that transpiration is even a side effect to photosynthesis, because when the stomata is open, due to various reasons, the idea about the stomata being open, which is controlled by the guard cells, is so that it's about to enable that carbon dioxide to come into the plant, because carbon dioxide is one of the crucial reactants in transpiration. However, when they open, they will also lose water vapor and oxygen during that process. It's important to note that for water vapor moving out of the stomata, that is considered to be diffusion. I mentioned in another video, when the water is not moving across a cell membrane, that is not osmosis, because the definition of osmosis is about how the water is moving down the water potential gradient across a partially permeable membrane. So in this case, if it's just simply moving out of stomata, then it's diffusion. So when the stomata is open to allow more carbon dioxide to come in for photosynthesis, water and oxygen also escapes, and that is why we say transpiration is a side effect to photosynthesis. Now we will have a look at the actual transpiration stream. So imagine if we zoom into the xylem, so that is my xylem vessel, and we look at the inside of it, we call the water molecules moving across. Whenever the water is moving through the xylem or in the cell wall, by the upper pass pathway, it relies on tension. And actually we say that there are actually two different types of forces that we need to notice here. One of them is talk about the hydrogen bond between different water molecules, and that is called cohesion. Keeping in mind that co relates to words like cooperation, etc. So about how different, two people working together. So cohesion is referring to that hydrogen bond between the water molecules that sticks them together. And the other hydrogen bond, which exists between the water molecule and the wall of the xylem itself, like that, is called adhesion. So think about adhesion, think about adhere. Adhere means to stick to something. So adhesion is talk about the hydrogen bonds between the water molecules and the wall itself. So these two are both the hydrogen bonds that are in the xylem that helps pull the water up. So in terms of the basic information about transpiration, just doing a very quick recap before we go into the next step, is transpiration is the loss of water vapor by evaporation through the stomata. And there are two things that are good about it, is that it cools the plant down to avoid any enzymes being denatured, and also it can cause a transpiration stream to actually happen to deliver the water and the ions to different parts of the plant. However, that means the plant is also losing water, which in an extreme case, plant itself can wilt. And the transpiration stream is simply referring to the pull of the water and the ions up the xylem because of the effects of transpiration to replace the water being lost in the cells and leaves. And if we look into the transpiration stream itself in the xylem, then we've got the two different types of hydrogen bonds. One is the cohesion, which is about the hydrogen bonds between the water molecules that stick them together, and adhesion, which is about the hydrogen bond between the water molecules and the wall itself. So this is transpiration. Now we'll have a look at the factors that affect transpiration rate and also how we can estimate it. So first we'll look at the factors that affect transpiration rate. Now we'll say if the transpiration rate increases, it's because of one or a combination of these things, right? So if the temperature increases, then the transpiration rate will also increase. And that is because there is an increase in kinetic energy of the water molecules due to the increased thermal energy. Because there's a higher kinetic energy, the water vapor molecules are moving around more, therefore they're more likely to actually move out of the plant through the stomata. If the humidity is low, then the transpiration rate would also increase. So if the environment is quite dry, then there will be more water loss outside because of how steep the water vapor concentration gradient would be. Notice in this case we don't talk about water potential gradient at all, because it's not about osmosis but about diffusion. So even though it's a little bit long, make sure you say water vapor concentration gradient. If there is more air movement, so if it's windy, then again it will also lead to an increase in transpiration rate. I think that's kind of straightforward, because if there is a lot of wind outside, then the relative humidity outside the leaf will also drop, therefore maintaining a steep water vapor concentration gradient. So kind of these 2 points are very much interlinked. Then the last one is about light intensity. If the light intensity is quite high, then the transpiration rate will also increase. And remember I said earlier, because transpiration is kind of like a side effect of photosynthesis. So if light intensity increases, then therefore more stomata will open to allow more carbon dioxide to come in for more photosynthesis. So again, these are the factors that would affect transpiration rate. Now the thing is, we can actually try to estimate the rate of transpiration. And this is one of the PAGs that you will need to know when you do this course. So in order to estimate that, we will use a potometer. Now the potometer is drawn here, so this is a setup of it. So imagine you've got a capillary tube, kind of like in this particular shape, and then on the capillary tube you will have a reservoir here, which can basically reset the air bubble position here, and you've got another bit here which it's got the fresh chute that is being installed there with the airtight gap there as well. And you try to get an air bubble into the capillary tube, and basically you just put the potometer into different conditions. So let's say underneath the sunlight, or at a colder or hotter area, or having it with a fan right next to it blowing at the chute, and you basically measure over a certain amount of time how far the air bubble would travel across the ruler. And then that would allow you to measure the water uptake by the plant. Now notice that we say that it is an estimation of the transpiration rate, not a measurement. Keeping in mind that the potometer, the movement of the air bubble will simply show how much water has been taken up by the chute. And the water can be used in different ways apart from transpiration. So it could be used up in photosynthesis, because again it is one of those reactants in photosynthesis, or it could be used to maintain turgor pressure of various plants' cells. So it could be used in these two particular functions, and then maybe if you got excess of it, then they might lose it by transpiration. So it is an estimation and not a measurement. Make sure you be very careful in terms of the use of words when it comes to answering exam questions. So let's have a look at this one in a little bit more detail, right? So we are going to let the air bubble travel across, and we're going to measure how far the air bubble has traveled in terms of distance, let's say in centimeters. And we use that distance to calculate the volume of water taken up by the plant. So usually people get a bit confused with this question, because they don't know how to convert it in terms of from the length to the volume of it. Keeping in mind that the capillary tube itself is a cylinder. So you basically need to calculate the volume of a cylinder. Imagine if we say the air bubble has traveled this much, so that is the distance traveled, and then you need to, the question will probably tell you the radius of the cylinder itself. So basically the volume uptake here is the volume of the cylinder, which is pi r squared times d. So you calculate the area, cross-section area of the circle, pi r squared, times the distance traveled, then you get the volume of water uptake. So in terms of exam questions, I would imagine that they might separate this into, it will be a bigger question, into subparts of it. So you might need to think about, they might ask you, how do you calculate the volume of the water uptake, what information you will need. Then you need to be able to say, okay, I need the radius of the capillary tube. They might ask you, why is it an estimation and not a measurement? Then you need to be able to explain that as well. They might even extend it to say, okay, what other factors, apart from the setup as given in the question, would affect the rate of transpiration? Then you need to be able to state these other factors and explain them. Another setup that is quite commonly used in schools now to replace that particular potometer will be this setup, because that potometer actually is quite a difficult setup to use. So actually most of the time that we use this one, which is simpler. So you got a beaker which will have the water in it as the reservoir to reset anything if you need to. Then you've got the fresh chute again attached to it. Then you got the capillary tube, and also maybe a third gap here, which will be with the syringe, which you can use to reset the water level in the capillary tube. So imagine here, you've got the water level starting here, and you set it at a particular condition. Then you basically, again, allow it to just run for a certain amount of time and see how much the water level would drop later on to, let's say, a new level. And that simply would be the volume of water that has been taken up by the plant during that time. It's easier to set up, and you don't have to try to get an air bubble in it. The graduated capillary tube will directly tell you the volume taken up by the plant during that time. So again, the quick recap of the second half. So we got the 4 different factors that affect the transpiration rate. Make sure you know what these 4 factors are, and also to be able to explain why and how they affect the transpiration rate. Then we talked about the potometer, which is a setup that measures the water uptake by the plant, keeping in mind that it only estimates the transpiration rate and not to measure it. We got, this is a classic setup. You need to be able to calculate the volume of a cylinder in order to calculate the volume of water uptake during that point in time. And then just so that you know that there are some other setup or methods to set up the potometer, which is simpler in terms of when you're doing this experiment. However, having said that, I would assume that most of the time in exams they will use this setup because, well, they will be able to ask you more questions on it and to specifically test on your math skills and if you remember how to calculate the volume of a cylinder. So keep that in mind. And that would be the overview of transpiration.